Moving on to our next speaker, we have Anthony Wong from the University of Toronto in Canada, and he'll be presenting TIMD4 and CCR2 mark-resident tissue macrophage subsets with conserved lifestyles across organs. Anthony, whenever you're ready, you can share your screen and take it away.
Hi everyone, good morning or good afternoon, depending on where you are in the world. My name is Anthony Wong, and I'm a PhD candidate at the University of Toronto in Canada. I'll be presenting my talk titled Identification of Resident Tissue Macrophage Subsets with Conserved Life Cycles Across Organs. So I'll be telling you a bit about macrophages today, and macrophages are innate immune cells that are present in all tissues of the body from the earliest stages of development, and they're essential for development and also maintaining homeostasis in both health and disease in their tissue of residence. And macrophages, like other immune cells, are important for fighting infection, and macrophages specifically have important functions in inflammation and antigen presentation. But macrophages are also involved in things like tissue repair and regeneration. So one example is that some animals, like salamanders, for example, have the amazing ability to fully regenerate tissue, even entire limbs, following amputation. However, if you were to deplete that animal of all its macrophages, this regenerative capacity is lost, suggesting that macrophages are important for tissue regeneration. And this is just one instance of that. But what's good for regeneration might be bad for cancer. So macrophages, like other immune cells, can help detect and destroy cancer cells, but they can also promote tumor growth in some cases. So macrophages have very diverse functions, and it's our recommendation that you consider them within the organ that you study. It's also important to consider the prevailing assumption that homogeneity can actually compromise precision. So macrophages from different tissues have been well established to have functional and transcriptional differences, but more recently, there's been a growing appreciation for the heterogeneity of macrophages that can be found within a single tissue or a single organ. And as an analogy, I like to think of a single tissue as containing different macrophage subsets or subpopulations like a fruit salad. And previous macrophage studies lacked consideration of pre-existing heterogeneity. So that's kind of like putting all those fruits into a blender and making a smoothie, to which those fruits are no longer distinguishable from each other. More recently, investigators have begun assessing each individual macrophage subpopulation in a given tissue or organ, because different macrophage subpopulations, like different fruits, like a kiwi versus a strawberry, for example, could actually be very different from each other. The problem is that each lab has their own way to define heterogeneity, so it's kind of like you can slice a pie 1,000 different ways. And this has led to a disparity and also a redundancy in the kinds of subsets that are being studied in the field. So the goal here is to address the lack of a unifying strategy to analyze macrophage heterogeneity by using single-cell transcriptomics, parabiosis, and phase mapping to determine whether different tissue-resident macrophage subpopulations could be organized and studied under a common framework. So we used a similar approach as what we published just over a year ago, in which we sorted the macrophages from the hearts of wild-type adult mice, and we submitted them for single-cell RNA sequencing to assess their gene expression. And for those of you that might not be familiar, in single-cell, each individual cell in a cell sample is measured for its total gene expression. And it's typically analyzed using dimensionality reduction analyses, in which the total complexity of gene expression of all your cells is reduced into a two-dimensional space in which each dot represents a single cell. And you can actually observe, or visualize rather, the cell clusters that form as a result of similarities in gene expression. So in doing so, in our bioinformatics analysis, we identified 3 transcriptionally distinct cardiac macrophage subsets. Those that we termed TIMD4 positive macrophages that uniquely expressed the markers TIMD4 and LIVE1, those that we termed MHC2 positive macrophages, which highly expressed MHC2 alleles like H2EB1, and those that we termed CCR2 positive macrophages, which uniquely expressed the chemokine receptor CCR2, among other genes, of course. And importantly, these different macrophage subsets were also distinguishable by flow cytometry, so we could actually observe them at the RNA and protein levels. So for instance, there are macrophages that express TIMD4 and not CCR2, and vice versa. And this double negative population here actually corresponds to the MHC2 positive macrophage subset that expresses neither TIMD4 nor CCR2. And so I used this single cell resolution to inform my fate mapping and parabiosis studies. And I extended these observations to several other organs beyond just the heart. And I asked the questions, do macrophages from other organs organize similarly by gene and protein expression? And number 2, what biological properties distinguish these different macrophage subpopulations from each other? And I explored things like ontogeny, life cycle, and turnover. So I sequenced macrophages from different organs, from the heart again, as well as the liver, and lung interstitial macrophages, which are all macrophages in the lung that are not alveolar macrophages. And so I sorted total macrophages for single cell RNA sequencing, and within each of the examined organs, 3 macrophage subsets resolved. And importantly, they could each be distinguished using the same key genes. So we were able to reuse the terminology as we did in the heart to name the subsets in the other organs. And here are the heat maps of the differentially expressed genes between each subset in each organ. And again, the takeaway here is that 3 macrophage subsets resolve within each of the examined tissues with distinct gene expression profiles. And therefore, importantly, they could be distinguished using the same core gene signature. So for example, TIMD4 positive macrophages in the heart express the same set of core genes as TIMD4 positive macrophages in the liver and lung. So next I asked a very simple question. Can I actually see these macrophages at the level of flow cytometry? So first I gate on CD45, which is ubiquitous on all immune cells. And then I gate on CD11b, CD64 double positive cells, which I utilize to identify macrophages. And from there, I stratify macrophages based on TIMD4 and CCR2 expression, which are on the y-axis and x-axis here respectively. And I assess these different organs at different stages throughout life. So here I'm showing 3 time points: an embryonic time point, an early time point, and an age time point. And in these bar graphs on the right, I'm showing the relative frequency of these different subsets to each other at the different time points throughout life. And again, the takeaway here is that these 3 macrophage subsets observed by single cell RNA sequencing could be distinguished by flow cytometry at all stages of life and development. And so it's one thing to identify that heterogeneity exists, but the more important question is to ask whether that heterogeneity actually has an impact on biological properties. And I have time to show one experiment today in which distinct macrophage life cycles were shown by long-term parabiosis. So for those of you that are unfamiliar, parabiosis is a surgical procedure in which the circulatory systems of 2 mice are surgically connected to each other. So the circulating cells in the blood of one mouse can pass on to the other mouse and vice versa. And in this setup, these mice were paired for either 6 or 10 months to assess the monocyte input to the different macrophage subsets, keeping in mind that circulating blood monocytes differentiate into macrophages once they enter into different tissues. And in this setup, wild-type CCR2 sufficient CD45-1 mice were paired to CCR2 knockout CD45-2 mice. Now, all monocytes in the blood express CCR2. So the neat thing here is that there's a unidirectional flow of blood CCR2 positive monocytes from the wild type to the knockout animal. And therefore, I assess the knockout animal for donor chimerism, which in the context of the knockout are CD45-1 cells in each of the different tissue macrophage subsets in each organ. And this was to determine the proportion of monocytes that could become monocyte-derived macrophages in each of these different subpopulations of macrophage in each organ. And I analyzed this data along several variables, but what was evident was the different life cycles that were revealed by subset, across subset. So the CCR2 positive macrophage subset in each organ was monocyte dependent, so they were nearly completely replaced by donor monocytes. The MHC2 positive macrophage subset in each organ was replenished by monocytes to a lesser extent and also in a tissue-specific manner. So, for example, the MHC2 positive macrophages in the liver experienced an increase in donor chimerism between 6 and 10 months, and it was the only group to show that. So all other groups showed no change in donor chimerism between 6 and 10 months, which suggests that there was a predetermined limit or ceiling for those monocyte-derived macrophages to take up. And finally, there was negligible monocyte contribution to the TIMD4 positive macrophage subset, which implied that they had a self-renewal capacity independent of monocytes. So these were present in their normal abundance and proportions, but they did not need monocytes to replace them. So they were able to stick around without the need for monocytes to replenish their numbers, which suggests that they were able to maintain themselves. So in summary, 3 transcriptionally distinct macrophage subsets exist in the heart, liver, and lung, and they can be distinguished by flow cytometry throughout life. This heterogeneity is correlated with conserved patterns in ontogeny, turnover, and life cycle between subsets across organs. And I showed one experiment showing life cycle today. And the take home is that our approach provides a common framework to study transcriptional and biological commonalities between macrophage subsets in the heart, liver, and lung. We also have data on the brain and kidney. And I'll continue to explore the implications of this heterogeneity to function and localization in my future work. And with that, I'd like to thank the Alphamin Lab, particularly, my supervisors, Sava and Sarah. And the bioinformatics work reflects a greater body of work with contributions from Sarah and Mira. And I'd also like to thank our collaborators, my funding sources, and you for your attention. So thank you so much, and I'm happy to take any questions.
Thank you, Anthony. That was great. If any of the attendees have questions, they can ask it in the chat. If not, Payam, do you have questions? Yeah.
Sure. Sorry, the sun is really starting to come out where I'm sitting in Boston. I do have great talk, Anthony. Really nicely done. One question I had was, I don't know if you saw this nice paper from Ron Germain's group a few years ago, where they looked at ILC subsets and they also had similar parabiotic experiments. So I'm curious, one of the things I'm curious about is if you did any S1P kind of antagonist experiments where you block the egress of the macrophages from the lymph nodes, and you're able to see if the subsets you define are really, tissue resident and how they're proliferating in their local context.
Thanks, Dr. Zarin, for your question. So I think I have come across that paper before, but I think the question, I guess, is mostly around the experiments that led me to conclude their different life cycles, or perhaps ontogeny or even cellular turnover. So to do that, in addition to the parabiosis, so we have wild type to wild type parabiosis, we have wild type to CCR2 knockout parabiosis to infer sort of monocyte contribution. And I also utilize a variety of genetic fate mapping lines, which I wasn't able to show today, but I utilize several different Cree reporter systems. And so one example of those fate mapping line that I study is the CCR2 Cree ER. So that's an inducible Cree at the CCR2 locus. And I mentioned earlier that CCR2 is expressed by all monocytes. So I can induce the expression of a reporter in circulating monocytes and transiently, and then I can observe where those labeled cells go and what they become. So I can observe, what macrophages they turn into, and I've observed that circulating monocytes can have the potential to become CCR2 positive macrophages, which is not surprising because they also express CCR2. To a lesser extent, double negative macrophages, but circulating monocytes contribute very, very little, if at all, to the TIMD4 positive macrophage subset. So that was what led me to determine that they did not require monocyte input in addition to the parabiosis studies. And I also studied ontogeny, so I use that line as well as the CX3CR1 Cree ER to pulse at different stages of development. So yolk sac hematopoiesis, fetal liver hematopoiesis, and I've determined that the TIMD4 positive macrophage subset and also the double negative macrophage subset in each organ is primarily derived by yolk sac progenitors, with also some contributions from fetal liver monocytes. But postnatally, they've received very little monocyte contribution. So I haven't done any studies regarding egress, but I think that would be interesting. And I think I would go there next. So thanks for your question.
Very cool. Such a productive PhD. I'm so impressed. Thank you for your talk.
Thank you so much for the opportunity. I can take any more questions.
Question for me, Anthony. So all these studies are, very elegant, and appreciate, it's a considerable amount of time in these mouse studies. I'm just wondering, your TIMD4 and CCR2, these are relevant to macrophage subtypes in humans?
Hi, Dr. Fish. Thanks for your question. So, so I didn't have time to go into detail today, but but a lab mate of mine actually does a lot of single cell, and she's actually assessed publicly available human single cell RNA-seq data sets from different organs.
Lost your voice.
I'm sorry. So I'll just repeat my answer. So a lab mate of mine actually has assessed publicly available single cell RNA-seq data sets from the human, from different human organs, from several different human organs that we've also studied in the mouse that include macrophages. And she's identified that the subsets that we observe in mouse are actually present in human tissues. They're harder to see during embryonic development, but in the adult tissues, those subsets are evident. However, they are distinguished using most of the same genes, but not all the same genes. So that was all to say, yes, we have seen similar subsets in human. Thanks for your question.
Thank you.
And then Payam was also wondering if you have any idea about macrophage subsets in the skin.
So unfortunately, we haven't really explored the skin. So we chose to examine organs based on several criteria. So membrane enclosed internal organs. Specifically, we examined the heart, brain, liver, lung, and kidney. I showed 3 organs today just for the sake of brevity. And we've analyzed different macrophage populations within those organs. We haven't analyzed ubiquitous tissues like skin and fat, but I think that's where we would be very happy to analyze those tissues as well. We're actually primarily a heart lab, so my PI is a cardiologist, so our expertise is in the heart. We just sort of like ventured into the other organs for this project, but I think I would be happy to study the skin as well. Thank you.
Awesome, thank you. Well, if any more questions come in, you'll be able to see them in the chat.
Okay, thank you so much.